Match question. A is chiasmata formation. Chiasmata is X-shaped structure and it is seen in diplotene. So A will be 3. A3 is in option 1 and 3. Next, B is common in both. So we are left with C4 or C2. What is C? C is synaptonymal complex formation. So synapsis is the event of zygotene. So this complex formation will also be seen in zygotene. So C will be 4. C4 is in option 1, so answer is option 1. Yes, this is the answer. So we took around 45 seconds to attempt this question. So this is very important topic. It is asked frequently. So we will go over it and understand each and every substage of prophase 1 of meiosis 1. So in this particular slide, I have picked up the statements from NCERT as it is given there, because a lot of questions are statement based or assertion and reasoning based. So my role as a tutor and educator is to help you remember those statements so that when it is asked in the question, you will be able to attempt it correctly and quickly. So let's begin now. Longer and complex than prophase of mitosis. What does this mean? Prophase 1 of meiosis 1 has events like recombination. So that this is the reason it takes longer time and hence it is complex compared to prophase of mitosis. Next one, the substages. So the mnemonic which you should remember is LZPDD. Leptotene, zygotene, pachytene, diplotene and diakinesis. So leptotene means compaction of chromatin just begins. So they are very thin structures. So normally we show chromosomes this way, thick structures, right? And with the sister chromatids. But here the compaction has just started. So chromatins are still thin and lepto means thin strands. So the main event, the highlight of leptotene will be compaction of chromatin continues. Next one, zygotene. Zygo is the term, the prefix means pairing. So the two chromosomes, homologous chromosomes. So remember one thing, homologous chromosomes in our cells are, one is from the father, which is called paternal chromosome. The other one is maternal chromosome. So actual exchange of the chromatids happen here. And this is how a new combination of chromosome will be created. And this is very important in bringing in variations in inheritance. So this is the reason prophase 1 is such an important event in the sexual reproduction, and it brings in variations which are inheritable. So for the recombination to occur, the chromosomes need to come close to each other, and that is possible only when the synaptonymal complex pulls them closer. So they actually align together, something like this. Very, very close. This way. Very close to each other. And this is the reason this is called tetrad or bivalent. So for zygotene, the terms which you should remember is synapses. What is synapses? The process of association or pairing. So in very simple English language, synapses will be pairing. And synapses is possible because of synaptonymal complex or synaptonymal complex. Then what else? The pair of synapsed homologous chromosomes is called bivalent or tetrad. So this is the highlight of zygotene. Next stage will be P. P stands for pachytene. Pachytene The tetrads are clearly visible. Appearance of recombination nodules occur. So recombination means the site at which crossing over of non-sister chromatids of homologous chromosomes occur. So the chromosome, this is the chromosome. So these are homologous chromosome, and these two arms are called non-sister chromatid arms. So exchange, the crossing over will occur between the non-sister chromatids. Okay, these two will be called sister chromatids. So chromatids which are connected by the same centromere are called sister chromatids, and chromatids which are on two different homologous chromosomes are called non-sister. So the exchange will occur between non-sister chromatids. And which enzyme is involved in this process? It is called recombinase. So actual cutting of the DNA on these two strands will occur. The chromosome structure will be cut and then resealed. So if I have to use a different color, you will have a chromosome. If this is the maternal chromosome and the red one is paternal chromosome, this is paternal. Okay, so after recombinase activity, what you will see, exchange of the chromatin strand. So this lower part will be transferred to the other chromosome, and it will look something like this. It will have a patch of, and this red chromosome will have a patch of blue chromosome. So this indicates that they have exchanged the material, and this is the reason now they are called recombinant chromosome, and the process is known as recombination or crossing over. So next stage will be, don't get confused between these two D. The first one is Diplotene and the last one is Diakinesis. So Diplotene, so since the job is done, recombination is over now, so the synaptonemal complex will also dissolve. So this is the highlight of Diplotene, dissolution of synaptonemal complex. But the two chromosomes remain attached at the site of crossover, and this X-shaped structure is called chiasmata. So where the exchange has occurred, these two chromosomes will still remain attached. So something like this. Okay. So this X-shaped structure is called chiasmata. So dissolution of synaptonemal complex and chiasmata formation are the highlight of Diplotene. The last stage is Diakinesis. This is marked by terminalization of chiasmata. So slowly the strand will slide over and the two chromosomes will separate. So they will be seen as separate entity at this stage. So terminalization of chiasmata is the highlight of diakinesis. What else is seen? The chromatin is fully condensed. So rest of the features are similar to mitosis. In prophase of mitosis, you will see chromatin is fully condensed, and then the spindle unit, they assemble on the poles, nucleolus as well as nuclear envelope disappears. So all the chromosomes are scattered around in the cytoplasm. So these are the highlight of each and every substage of prophase 1. So all the best with this topic, and thank you so much for watching my video.